Hi everyone. So I'm gonna try to finish up a little bit of the explanation of Unit 2 thinking practice. So we left off in calculating water potential. Okay, so hopefully that made a lot of sense. Remember that the calculation of water potential is solute potential and pressure potential. Pressure potential will only be relevant in a question if they tell you that the beaker of within where the solution is held is a closed beaker, so that it doesn't allow any like volume change, and that means that there could potentially be pressure once water starts moving. And it's also relevant when you're talking about plant cells, because since a plant cell has a cell wall, it can only take in that a certain amount of water. After a certain point, the cell wall is going to start putting pressure against the entry of water, and no more water is going to enter. If we're talking about a cell that contains a higher concentration of solutes in cell, a plant cell, that contains a higher concentration of solutes inside that cell than on the outside, water is going to want to move in, but it will only be able to move in up until a certain point when the cell wall is going to start exerting pressure on the cell so that water doesn't move in anymore. And that is what pressure potential is. If there is no mention about a plant or no mention about having a closed beaker, then you can assume that this is zero. Okay, and then water potential is simply the same thing as solute potential. So what I wanted to show you, remember always that water potential will move from water will move from an area of higher water potential to an area of lower water potential. Okay, always remember that. But one thing that you can always use to understand water potential is that remember that since water potential has to do with solute potential, if a solution has a lot of solutes, that means that it has a lower water potential. Why? Because water potential is the potential of free water molecules to move. If there's a lot of solute, there's a lot of water molecules that are attracted to that solute, so they're no longer free. So you have decreased the water potential because you have decreased the amount of free water molecules. How could I have answered question C of in which way will water move without actually doing any of this math? I could have answered it just based on logic. If it tells you that in one side of the membrane you have 0.1 moles of glucose, and on the other side of the membrane you have 0.1 moles of calcium chloride, but the calcium chloride ionizes into three new solutes, then what you can think about is in one side you have glucose that doesn't ionize, so it only produces one solute, and on the other side you have calcium chloride, which ionizes and produces three solutes. That means that on the one side you're going to have 3 times as many solutes that on the other side, okay, on the glucose side. So since water potential decreases with the amount of solute, the side of the solution that contains more solutes are going to have less water potential than the side of the solution that contains less solutes. So in this case, the glucose solution is going to contain less solutes than the calcium chloride solution. And the calcium chloride solution is going to attract the water from the glucose solution, so there's going to be that water movement. So hopefully that makes sense. You could have simply known how water moved without doing any of the math based on the amount of solutes. 1 versus 3 times as many. Okay, let's go into proteins. So this was super easy. This question everybody got right, so I'm not going to go into it that much. It was where would the hydrophobic R group of a protein within the bilayer be? Well, if it's hydrophobic, it needs to be in the hydrophobic region of the tails of the phospholipid. The hydrophilic R group, it would have to be in the hydrophilic area of the heads of the phospholipids that comes in contact with the aqueous solution, the water-based solution, outside of the cell and inside of the cell. Okay. This question was a little bit difficult, so hopefully you guys were able to understand it. First of all, this has to do with action potential. So if this is a neuron, the action potential is the change of voltage from the inside and outside of the cell so that a signal can be carried on. So the inside of the cell is usually at minus 70 millivolts of voltage, okay, and that is your stable resting potential. And if you want to have an action potential, you need to change that voltage. So you actually increase the voltage a whole bunch until it passes an actual threshold and carries that signal. And then you need to go right back into the resting potential to wait another signal, to wait another action potential that you need to send. Okay, so to be able to do that, first you need to think about three different characteristics. One is just your sodium potassium pump, and that is used simply to maintain your resting potential. Then you have a sodium channel, voltage gated channel, and that is actually to do the depolarization, so to increase the voltage inside the cell. Then you have a potassium voltage gated channel, and that is to do the repolarization, or to decrease the voltage within the cell so that it can go back into a resting position. So if you want to see it in this diagram, I think this diagram is pretty good. Just to maintain your resting potential, remember you have this protein, this a sodium potassium pump, where constantly you are pumping 3 sodiums out and 2 potassiums in. And since both of them are positive ions, you can see that you are changing the charge between the outside and the inside, because you are sending more positive ions out than the amount of positive ions that you're sending in. So there's a difference between one positive ion. So on the outside, there's going to be a more positive charge than on the inside, and that's just your resting potential. Once you're trying to use an action potential, you want to depolarize. That means that you want the inside to start becoming positive. And the way that you can do that is that you can use a sodium channel, voltage gated channel, and that just means allowing some of the sodium that you pumped out to come right back in. Okay, and as you're allowing the sodium that is positively charged to come right back in, you're allowing a positive charge inside that cell, okay, in the cytosol of that cell. So once you reach enough positive charge on the inside of the cell, the action potential will allow to carry a signal through those nerves, and it will carry it up until the point where the action can be made. So moving a skeleton, for example, moving your arm, for example, has to do a lot with this. Okay. Once the action potential has been fulfilled and the signal has been carried, now you want to repolarize. You want to go back to your resting potential. And to repolarize, that means that you need to get the inside to be negative once again. And to get the inside to be negative once again, what you're going to do is you're going to expel or let go of some sodium onto the outside, and this is through a voltage gated channel of sodium, potassium, and it allows it to exit. So potassium exits, and since some potassium is positive, then a lot of your positive charge is exiting to the outside, so it allows you to become negative once more. And to regulate this to get to exactly minus 70 millivolts, then you get a potassium pump once more right after the repolarization. The potassium pump works once more so that you are able to get to that minus 70. So if you want to look at that in this, here you would have your resting potential, sodium potassium pump. Here you would have your sodium voltage gated channels open. Here you would have your potassium gated channel voltage channel open, and here you would have once more your potassium pump working to get that resting potential of minus 70. So hopefully that makes sense with all of these questions. Okay, in this question, what was super important is that it was asking you to tetrodotoxin is a neurotoxin that blocks sodium gated voltage channels. How would the function of the neuron be altered in the presence of the toxin? A lot of people said that you wouldn't be able to do this depolarization, which is completely true, but you didn't answer how would the function of the neuron be altered. And what the function of the neuron would do is that it wouldn't be able to send that signal, because you need this change in voltage to be able to send the signal. So you wouldn't be able to send the signal, and that's why neurotoxins are so crazy dangerous. Everybody got this correct. Everybody got this, that it was lysosomes, because you're not able to break up these very large complex lipids, so they become clogged, and it means that your lysosomes are not working. In a little bit of in the lung-free response, a lot of people got this right as well. The only thing that you need to make sure is that time is your independent variable. So this has to be in your x-axis, in your horizontal axis. This concentration needs to be in your dependent variable. It's your dependent variable, so it had to be in your y-axis. Okay, and in your graph, you needed to include the 0% concentration, the 1%, the 5%, which is what you're seeing here, and the 10%. You needed to include all of those percentages. And the 10%, a lot of people put it very close to the 5%, when 10% should have been double the concentration. Okay, so you should have adjusted your y-axis to allow to graph that 10% as double the amount for the 5% sodium chloride. Okay, and farmlands located near coastal regions are being treated by encroaching seawater seeping into the soil, so the seawater is going into the soil. In terms of water movement into or out of the plant cell, explain why seawater could decrease crop production. Include water potential in your answer. A lot of people did not include water potential in your answer. So the idea was seawater is entering the soil. Seawater has a super high concentration of solutes. Since it has a high concentration of solute, it has a low water potential. The water inside the plant cell might have a higher water potential, because plant cells do not have a high concentration of salt. So the water inside the plant cell is going to want to move, because it has higher water potential, to the outside close to the seawater that has a low water potential. In this way, the plant cell could become dehydrated and could eventually die. So hopefully that made sense. And yeah, check out your answers.